This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 520, recorded on November 16th, 2018. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me here in New York City, Dickson Despommier.
Good afternoon, Vincent.
We're just practicing his name, Despommier.
Yeah, well, that's true.
In the aftermath of a small storm, right?
We had our first snow.
Our first snow of the year.
That's right.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's great to be back.
Were you not here last week?
Right. You didn't know.
Oh, you were at Hamilton. You were at Hamilton. That's right.
Right. I went to Chicago.
We talked about you.
Oh, okay.
Briefly.
My ears were burning.
We said that we're having fun, and then I said I think Kathy's having more fun. Was it good? It
was great. It was really good. I want to see it again.
Oh.
All right. Did you see it, Dickson?
Not yet.
Neither have I. All right. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. I'm very jealous of you, Kathy. I really want to see Hamilton.
Yeah, it's affordable and accessible in Chicago.
Yeah, not true here.
No.
Still not true? Yeah.
No, it's expensive.
No, not true.
It's pretty expensive.
It's going to be twice as much when it comes to Detroit for a 3-week run.
Really?
So I'll go back to Chicago to see it again.
Interesting.
Doesn't the guy live nearby here?
He does. He saved a local restaurant, as a matter of fact.
What's his name?
Lin-Manuel Miranda?
That's the one.
He saved Coogan's. He saved Coogan's, right?
He did. His family eats there all the time, and they were going to close it for whatever reasons. And he heard about it and through the grapevine and actually got a visit with the president of the hospital corporation here and convinced them otherwise, that this is a worthwhile place to save. All of the people who worked at the restaurant, however, had made other plans because they thought they were closing. So now they were scrambling around for places to live and that sort of thing because they had actually sold their apartments.
This is a restaurant where Dickson has his own personal table.
He does.
Wow.
He does.
Ooh.
And there's a wine on the menu selected by Despommier.
Yeah, it's not there anymore.
Some of your art is on the wall.
That's true.
Oh.
That is true.
No, I'll have to go there.
Oh, we'd love to take you.
Next time you come to the TWiV studio, I'll take you there. I'm sorry to say the food isn't very good, Dickson.
Well, it depends on what you order.
It's Irish food, after all.
They've got the best macaroni and cheese in town, I can tell you that. Also
joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
All right.
We got 70 degrees, 69 degrees. That's 20 Celsius, clear blue skies.
You're just going to rub it in? Just going to rub it in, aren't you?
And from Western Massachusetts, Alan Dove.
Good to be here. And, Coogan's, I guess I was last there about a year or two ago, and the burgers were quite good.
They are good. They are good.
I had remembered it from graduate school as a place to go tie a couple on before going out for the rest of the evening. But,
something like that.
They should serve Miracle or Wonder Burgers, whatever they are.
The wings are good too, by the way. There's a lot of good things now. I think you'd change your mind if you peruse.
What is the name of that vegetarian burger? The Impossible Burger.
Impossible Burger.
Yeah, that's it.
Which is surprisingly good.
The pseudo burger.
I heard.
It's really worth trying. Yeah.
Yeah, Pat Brown, inventor of many scientific techniques at Stanford. He was a Stanford professor, took a leave of absence and founded this company.
Wow.
He wanted to cut down methane emissions.
Really?
Yeah.
Yeah, it's very cool, except they still have the same number of cows in the country. But.
Oh, that's true.
Well, but burgers are a huge a huge way that people consume beef. And if you can find a substitute for that, that's vegetable based, and it is a credible patty of beef. I mean, it's actually, it's not the absolute best burger I've ever had, but it's certainly not the worst. And it's, I think if fast food restaurants went to that, it would be an improvement.
Well, White Castle is, right?
Yeah. Yeah, that's a step in the right direction. So welcome to This Week in Burgers.
This episode is brought to you by the graduate and postdoctoral programs at the Department of Microbiology, Icahn School of Medicine at Mount Sinai, composed of over 20 virology labs, all centralized in one building in the heart of New York City. This department is a perfect fit for anyone with an interest in pursuing virus research. The department is presently looking to recruit prospective graduate students to apply to the program by the December 1st deadline. Interested postdocs are also encouraged to contact faculty of interest. For more information about the department, please visit bit.ly/micro MSSM. That's all one word, MICROMSSM. What is the .ly domain?
Libya?
Libya?
Yeah.
I thought so.
Okay.
.ly domain.
And the domain has been much more stable than the country.
Interesting.
Libya. Yes, it's Libya. Does anyone know what .tv is?
Tuvalu?
Tuvalu, yes. Okay, we don't need to go down this rat hole. Sorry. University College London wants you to know about their MSc program in molecular biology and pathology of viruses. This course offers an integrated approach to understanding the nature of viruses, their role in disease pathogenesis, with an emphasis on understanding of these processes at a molecular level. The objectives of the course are to equip those intending to pursue a career in academic or industrial research with the required knowledge and skills, and to provide a solid foundation for those who aim to go on to study at a PhD level. More information is at imperial.ac.uk/study/pg/medicine/molecular-biology. Wow, it's a long one. You ought to get a Libya thing. The last plea to everyone to subscribe to my YouTube channel, youtube.com/PROFVRR. And I don't think we're going to make 25,000 so that I can apply for a grant, because we're not even at 17,000.
You're getting close.
But try. I think we picked up 1,800 since I began pleading. It's kind of pathetic, but that's fine. For sale is Susan Nassif's virus comic book, Adventures of the Regaches, Volume 1. Vaccines help support her work. We'll put a link in the show notes. It's a short one, but it's too hard, too complicated. And I don't know if this is new, but Paul Dupre last week sent me a virus emoji.
Yeah.
Are these new?
That's I don't know what the status is on Android or other operating systems, but on iOS for Apple devices, they just updated to the latest one that gives you these new emoji. And I found out about it because my wife texted me a bunch of the new emoji, which of course come out as question mark blocks until you update your operating system.
Oh, even if you do a screenshot, it would do the same thing?
No, screenshot. If you do a screenshot from a device that can display them, then you'll have the images.
Okay, so I'm doing that right now for those of you that can't see it. I just
there's a virus, there's a mosquito, there's a petri dish,
test tube, DNA.
And they do show up on Androids.
All right, I just pasted
I figured there'd be.
I just pasted it in.
Cool.
Got a what's the flasky called? Oh,
I don't know what that's.
Florence flask.
No, it's
Erlenmeyer.
No, they call
it's a Florence flask. The round bottom flask is Florence.
No, it's not.
Oh, because it's on its side. Okay.
Yeah.
Corel.
No, it's a
It's like a old style
alembic.
Alembic.
It's an alembic.
Right.
Boy, you guys are way ahead of me. Wow.
If you go to the emoji, I don't know that. They told it.
It depends, because for me, the microscope, telescope, and the flask do appear correct, as I put in my screenshot.
Weird. It's very weird, right?
But not all of them. But on my phone, they would all look okay.
So you have probably the latest iOS on your phone.
And not on my computer.
Yeah, that's probably why. Anyway, the virus is... If you search for virus, it'll come up, but it also says microbe under the picture, so I guess it's multifunctional.
Yeah, but if you're texting and you type virus, then it pops up.
It'll pop up. Yeah. And the petri dish has mold on it. If you blow it up.
It does. I hadn't thought about that.
Homage to Alexander.
So the word emoji, it was derived, it seems to me, to describe symbols that evoke emotions or communicate emotions.
Mm-hmm.
So what emotion is communicated by a virus?
Panic.
That's a
or
good point.
DNA,
or DNA,
or a
yeah.
microscope.
Yeah.
So the word emoji has an expanded meaning now, I guess.
Maybe they should have a picture to represent the word.
We have some follow-up. Bob writes, I think Rich should take this one. Rich, take that first one.
Bob writes, near the end of the podcast when Rich Condit was talking about the voyages of James Cook, I wondered if he had read the book by Richard Holmes, The Age of Wonder. Any book by Richard Holmes is a treat, but this one is special for those who love science since it details an age in which science was a fascination of just about everyone in the world. The book begins with Joseph Banks in paradise. Banks was the official botanist on the H.M. Bark Endeavour, commanded by Lieutenant James Cook. That's the one that made the first voyage, Cook's first voyage around the world. It goes on to cover many more topics, all of which are fascinating. Regards, Bob. Well, thank you, Bob. I read this a couple of days ago for the first time and immediately went electronically from my Kindle to the library and discovered that the book was available as an ebook and downloaded it. And Joseph Banks and myself have just had our time in Tahiti and are off to the rest of the world. So I'm well into it. Looks good.
Mikhail writes, hello, dear TWiV hosts. Traditionally, longtime listener, second-time writer. Few notes about the conversation in latest TWiV. 1. Trigeminal nerve is cranial nerve 5. It cannot be 13, as Dr. Despommier vaguely suggested. There are only 12 cranial nerves. I don't think you vaguely suggested it. You said 13.
I thought I said it might be 13. But
I see.
So this,
Did he vaguely suggest it?
Vaguely. He did. He had a hell of a nerve suggesting it. You can see in my pick of the week that I've done a mea culpa.
Number 2, ganglion, plural. Ganglia is the accumulation of neuronal bodies rather than nerves, in reference to Dr. Condit's comment. Oh. These bundles of neuronal bodies are called ganglia in the peripheral nervous system, and they are nuclei in the CNS. For example, dorsal horn ganglion can be found in the dorsal root of a spinal nerve. This is excellent. Number 3, prevalence of herpes simplex 1 infection increases with age, as Dr. Condit noted. To corroborate this notion, I attach links to 2 papers with the serological survey data obtained in Slovenia and China. These data also suggest that HSV1 seroprevalence is much higher than that of HSV2. Thank you for your service to science. Driving around Cleveland is much less stressful with your podcast in the car audio system. Right. Mike is a PhD assistant professor at the Lakeland Community College in Ohio.
Oh, cool.
And I just love that Mike properly says these data.
Indeed.
And most people seem to say this data, right?
Yeah.
Yeah, that was an obsession of mine running my laboratory. People, there are a gaggle of people who came through the Condit lab who understand that data is plural.
Yeah, are plural.
Data are plural.
Data are plural.
These papers are quite interesting. The one from China, herpes 1 seroprevalence, 92% overall in 2,100 participants in the survey.
Wow.
But 61% among children 5 to 9 years old, 90% 25 to 29 years old, so big jump there, and nearly 100% if you're over 40 years old. And it's less for HSV2. So HSV1 is quite high. And it increases pretty rapidly, pretty dramatically.
Do we know why?
Well, you just
more exposure.
More exposure, yeah. More likely that you're going to get some...
You live longer, you're just more likely to pick it up.
Some saliva.
Okay. All right.
Kathy, can you take the next one, please?
This is the one from Ruanne and Michelle, who write, Dear TWiV, HSV1's apparent association with Alzheimer's disease has been the hot topic in our lab the last few weeks, and we thoroughly enjoyed the episode addressing it. We would like to add a few thoughts from our perspective in clinical virology. The suggestion that lytic viral replication is the etiology seems, at least anecdotally, doubtful. The detection of HSV DNA in cerebrospinal fluid is exceedingly sensitive and specific for HSV encephalitis. such that when detecting it, we can generally provide the diagnosis with some confidence. For context, a diagnosis exclusively from a lab test is uncommon, but the correlate clinically proviso that goes out with all our reports should really be replaced with a bag of acyclovir when it comes to a positive herpes simplex virus, cerebrospinal fluid, PCR. HSV encephalitis is also not a mild illness which may be dismissed. It comes with a high mortality and invariable neurological sequelae for survivors. So significant lytic replication, we doubt. The data showing HSV's Alzheimer's association is there, however, and needs to be understood. On the one side, focal contained areas of replication can certainly not be excluded with ease, but one has to question which way the train is going, as the TWiV team mentioned. One perspective might be the creation of focal zones permissive to HSV replication, possibly through innate immune dysregulation secondary to Alzheimer's pathology. This particularly is an attractive thought, considering the association of HSV encephalitis with defects in toll-like receptor 3 signaling shown by Zhang et al. In 2007, with subsequent further investigation by other labs. Further, an explanation for the data regarding the negative association between Alzheimer's and acyclovir use is also interesting. One might be tempted to think that it could be explained by activation of the HSV thymidine kinase gene in stressed neurons without progression to lytic replication. This may result in targeted genotoxic effects resulting in apoptosis from administered acyclovir in stressed cells, which may have acted as paracrine immune dysregulators driving pathology. This principle has already been extensively explored in cancer gene therapy, with cancer cells being transduced with a thymidine kinase gene and ganciclovir subsequently being administered to kill the cancer cells. Acyclovir triphosphate, post-thymidine kinase and cellular kinase activity, does favor viral polymerase, but if not all viral genes are expressed, parentheses polymerase gene, cellular polymerase may incorporate it with greater frequency. These ideas may, of course, be totally wrong, but we think a lot of investigation will need to happen and many people will need to be wrong before we understand the true nature of the apparent HSV-Alzheimer's association. P.S. Cape Town is a great place to come do a TWiV episode, especially on HIV vaccinology.
Yeah.
Brianne and Michelle, who are residents in clinical virology at the University of Cape Town.
I'll be in Cape Town in January.
Really?
Oh.
It's a great time.
It is.
Oh, that's right. You do that thing, that trip.
Yeah, I take students abroad to South Africa over winter break every year.
Yeah.
Lucky duckies.
So the idea, lytic replication, we doubt, was kind of what we thought last year. And so I'm happy to see that some other people have the same ideas. And this idea that some viral genes are produced is interesting also. We suggested that. So nice to hear. I would have loved to hear what they think about the clinical trial.
Well, I really like this bit about acyclovir because we talked about the notion that the drug may be doing something other than what we expect it to do, which is to tamp down the herpes infection. And this is really interesting. For the lay folks out there, the idea here is that the way acyclovir works, without going into detail, is that a specific virus gene product activates the drug, in effect. Okay, but once activated, it can have toxic effects not only on the virus, but on the cells. And so even if the virus weren't replicating, if it made this gene, thymidine kinase, which is not improbable, the latency is not black and white. You get little bits of gene expression here and there. It could activate the drug and ultimately kill the cells. Okay, that's very interesting.
Render certain cells sensitive to killing by acyclovir.
Yeah, right. So that's interesting.
Yep.
It is. And the idea that those cells were sort of dysregulators that were driving other problems, with the pathology is also really interesting.
Very cool. Yeah. Glad to hear they're listening in Cape Town. The American Society for Microbiology is proud to support Outbreak: Epidemics in a Connected World on display at the Smithsonian National Museum of Natural History, and is going through May 2021. And unlike Hamilton, it will not break your wallet to go see it.
No.
Because it's free. Here at TWiV, we're excited to see the topic of zoonotic viral diseases getting such a public platform. As a tie-in, we'll be discussing the latest information on one of the viruses featured in the exhibit in a special segment each month. Well, we haven't been quite monthly. We've only done one previously on Ebola.
I thought we'd
virus.
done two.
Did we do Nipah? We did Nipah and Ebola, but I think we skipped a month. This month's segment is on influenza virus. If you're in the Washington, D.C. area, be sure to go see the exhibit. It's free. And don't miss TWiV 501 Outbreak. So the exhibit's all about zoonotic viruses. So you may ask, why is influenza virus in it? Well,
oh, come on.
That's because some people may wonder, flu, right? They think of flu.
Even I wouldn't have asked that.
But you are a scientist.
Yeah, and you've been hanging around with
I have.
TWiV for 10 years.
Yeah, well.
10 years of TWiV.
10 years of TWiV.
So the reason is because all human influenza viruses came from some sort of animal at some point. Many of them are now human viruses. They transmit from human to human, but there's a huge animal reservoir of influenza viruses in all sorts of birds, for example, both aquatic and terrestrial, and also many other animals, pigs and what else, whales and seals and many other animals. So, and periodically the genes of these influenza viruses that infect those animals reassort with human genes and produce a brand new pandemic strain that infects many millions of people. So it's a classic zoonosis. And, you know, this emergence of a reassortant, a human animal virus reassortant, has happened multiple times. 1968, 1957, 1918, of course, etc. And so that, together with year-to-year antigenic variation, is why we have to keep getting influenza vaccines. And I thought we could focus on an interesting issue, which is, you know, right now we have to get a flu shot every year, but many people are talking about a universal influenza vaccine, one that you would get maybe once or a few times in your life and could protect you against all isolates. And in fact, the National Institute of Allergy and Infectious Diseases has just published a strategic plan for developing such an influenza universal vaccine. And of course, Tony Fauci is a big proponent of this idea, and he has written an article about it, the strategic plan for the National Institute of Allergy and Infectious Diseases. And he's got, it's a nice article to look at because they basically say, what are the characteristics of a universal vaccine that we would like? And they list 4 of them, which are not yet on my screen, so I can't tell you.
It should be at least 75% effective, protects against both group 1 and 2 influenza A viruses, have durable protection that lasts at least one year, and be suitable for all age groups.
There you go. And so they're funding big time. In fact, this year's NIH budget increase, $2 billion. A good fraction of that goes towards research on universal influenza virus vaccines.
Yeah. Now, one could legitimately ask, why weren't we working on this in the first place? And the answer, of course, is we have been, that people have been trying for a very, very long time to find universal flu vaccines. That would have been the first choice rather than these annually updated ones. But it turns out to be really hard.
People have been working on it for a long time. And there's a wonderful... by the way, if you're wondering about flu, many people, of course, still do not get flu vaccines, but the last season was quite serious. Yeah. We can put a link to the CDC site, but many, in excess of the normal mortality that you would see for influenza, which ranges from between 3,000 or 49,000 deaths a year just in the U.S. So this is certainly a medical need.
Yeah, so, people kind of shrug off flu, but it's this is a killer.
It is a killer.
All right? It kills a lot of people every year.
Yep. So
just to be clear, it's not all really, really old people who are going to die of something else soon anyway. I mean, yes, the elderly are particularly susceptible, but a lot of the people who die of flu each year are kids. And, of the kids who die of it, the overwhelming majority of them in most years were not vaccinated.
Right.
Dickson, did you get your flu vaccine this year?
You want me to tell the truth? No, I have not.
Oh, why?
There have been several times when we were false started.
Yeah.
Got diverted from attention, et cetera, et cetera.
But you can still get it.
We're going to get it this week. I promise
In fact,
we will get it this week.
Mm-hmm.
I heard that, or I read somewhere that this is actually the ideal time so that your immunity will last through the entire flu season.
Oh, good.
Right. Yeah.
Oh, good. Okay.
Not in
so
August,
you can rationalize it. Yeah.
No, I've been
You waited
angsting, and I was hoping you wouldn't ask me that, actually.
Good move. It's okay.
So what if I got it like 2 weeks ago?
Good.
Still did.
Right.
Yeah.
If you got it 2 months ago, it's still
promise.
All right. I was at a meeting here, and they gave it to us at this meeting.
Oh. Yeah.
Nice.
It was a biosafety committee meeting.
Oh, that was a good idea. They stuck it to you, Em.
They stuck it to me. The Scientist has a good article summarizing the progress on a universal influenza vaccine, UIV. So let's talk about a couple of them.
Wait a minute. I want to make one more comment about Fauci's paper. What I found interesting about the paper is that, when I think of making a universal flu vaccine, I immediately go to the actual process of making the vaccine. But this effort to the strategic plan to make a universal flu vaccine, 80% of it at least is research on flu transmission, pathogenesis, genetics, all kinds of stuff. Okay, so it's I found that really interesting and quite appropriate. It's not just let's make a vaccine; it's let's understand the virus in all ways we can and use that information to make a vaccine.
And it's clear from the efforts that have been made so far to develop a universal vaccine for decades. Just rushing in and saying, okay, we're going to do a universal vaccine. That's clearly we don't know enough. We've tried that, and it's not as simple as that. We really need to know a lot more, especially about the immunology of this virus.
Exactly. As we've talked on this show many times before, correlates of protection, right?
Right.
How is it that you become immune to something? And we measure a lot of things, and we assume that those things are important if they go up when you're infected. But it's been shown time and time again that we're pretty naive about this. And understanding the immunology better is important.
And what we see with flu vaccines is it's pretty easy to stimulate antibodies against the hemagglutinin and neuraminidase, the H and the N components, and that's usually what we're kind of aiming for with flu vaccines. So if you get immunity to an H1N1 virus, you're immune to the hemagglutinin and neuraminidase on that virus. But then, some H3N2 comes along and you're not immune to it. That's kind of the conventional approach. And that's the problem because these reassortants occur, as Vincent described. And now you've got a new virus circulating, and that's why you need a new vaccine every year.
Are we clear, by the way, as to why none of the antivirals work very poorly against this virus?
You want to know why they don't work better?
Yeah. Mm-hmm. I do. Like Tamiflu, for instance. You keep hearing about it, but it doesn't seem to really do much. So it doesn't save lives, apparently.
Well, it tamps down it tamps down the spread. Okay, which is, it's not like it stops the virus in its tracks.
Yeah. Well, why can't we do that?
Not only that, one of the problems is that by the time you show up with symptoms, you're
too far gone.
You're pretty far down the road.
Does a lot of the damage then, is it innocent bystander type of thing? When the cell dies, it releases a lot of stuff that then affects a whole bunch of other cells that have nothing to do with the virus itself?
There's some data showing that, yes.
Mm.
And of course, you get the whole cytokine storm going on, and the response, and there's a lot of there's evidence that a lot of what drives flu mortality is that plus secondary infection, and secondary infection is obviously a huge issue here.
Yeah.
And then there's the whole drug resistance, to antivirals. And if we had a cocktail of things for flu that affected different processes in the virus life cycle, different enzymatic things that are unique to the virus and not in the cell, we might have a hope of being able to deal with the disease better.
Remarkably, and I hate to say this, but it's a political situation, because when the AIDS epidemic first came out, how many people have died from AIDS over the years that it's been around versus how many people have died from influenza for the same number of years? It sounds like more people have died from influenza.
I don't know that we have those statistics at our fingertips.
Yes,
but I mean, you're up to, let's say it's even. Let's just, for the sake of argument, say it's even. So HIV right now has a wonderful triple cocktail of antivirals that seems to work.
So we've had 35 million AIDS deaths.
Yeah.
Right.
Depends on how far back you take the flu statistics, but I'm pretty sure they
No, for the same number of years.
For the same number of years, yeah. Okay.
Yeah.
Well, I don't know what the global flu is. The numbers we cite
3 million. It says in this
3 million deaths when?
It says right here.
Yeah, in your
blurb
note here, it says between 300,000 and 500,000 deaths globally per year.
Oh, right.
Yeah.
Right. It's less.
It's less than AIDS.
Yeah.
Yeah. And the important thing to remember, though, is that we could prioritize viruses all we like, but the viruses don't care. And a lot of what has driven progress with HIV is the ways in which we can target that virus. And it turns out that we can make reverse transcriptase inhibitors. We can make, these compounds that are pretty effective against it. And so, we've got good antivirals now against HIV, which was the result of a lot of research. It's not clear that we'll be able to make good antivirals against flu because it's a totally different virus, and we're not guaranteed of necessarily being able to get there.
But you have strange proteins produced here that we don't have, like hemagglutinin and
sure. There's
the neuraminidase. So
there are targets, but people have been trying to hit those for a long time, and we've got Tamiflu, and we're still debating.
Is that what it does?
Whether that works?
Inhibits both of those things.
Well, there's a new one now that hits the endonuclease,
right?
Which
Tamiflu is neuraminidase.
So there's a new one. And the problem is you can make antivirals that inhibit virus replication in cells, but you only have a short time.
Yeah, sure.
Yes.
Whereas
Whereas HIV, you got your whole life to do it, right?
Yeah,
and so that is a big difference.
That's a huge difference. You're
and as Rich said, if you were a day into replication already, then the impact is going to be much less.
Right.
Now, if you want to convert flu to a chronic lifetime infection, then yes. But I don't think you want to do that.
And on the other hand, we have flu vaccines, and we can complain about their efficacy and but they exist, and they're distributed and given, and they do have an effect, and they keep people out of the hospital. We have no HIV vaccine. So it turns out it's easier in a lot of respects to vaccinate against flu. We just can't get a universal vaccine until we take a different approach, and that's, I think, what we're going to talk about next.
Trying to find the name of the new.
I was trying to do the same thing. It was in our paper the other day, Vincent.
Yeah.
I can get the old drug numbers and that kind of stuff, BPR3PO128, but that's
Baloxavir.
Baloxavir.
Yeah, or
it only works in Mississippi?
Yeah, or Zofluza, right? It's both wonderful names.
Yes.
Baloxavir marboxyl.
Sounds like a Dr. Seuss story.
Oh, boy. Anyway, that was licensed a while ago in Japan and just now in the U.S. And I think it's going to have the same issue. By the way, Dickson, you said, does it prevent death? That's hard to measure.
Right.
Because for obvious reasons, right?
I'm asking for the general listener right now. Basically.
As a matter of fact, the CDC site is quite circumspect about talking about the exact number of deaths every year due to flu, because it is hard to measure.
Yes.
A lot of people die with flu rather than from flu. Yeah.
Right. And we have the same problems when we talk about vaccine efficacy. The vaccine was only 40% effective because we measured that people got the virus. Well, did they die? Would they have died if they hadn't had the vaccine?
Sick. That's you're right.
So, but what we'd prefer to do is obviously not have to immunize people every single year with a brand new vaccine.
Right.
Exactly.
If you had a flu vaccine every 10 years, I think more people would get it and fewer would complain. Every year is just hard. Even though everywhere you go, you got signs there. Flu vaccine available. And
right, but that's
covered by your insurance.
Of a huge logistical network that is very difficult and expensive to maintain. And it'd be great if we didn't have to do that.
Yep.
Yeah.
I've been surprised at how many of my non-scientist friends are confused about why you have to get a flu vaccine every year.
Yes.
And I've had to have do the explanation of, no, the virus actually changes pretty often. So I think that having a broader understanding of that, or having a vaccine that actually worked against multiple viruses would be very helpful for people.
Right.
And the other thing is, herd immunity, obviously being what it is, if everybody gets vaccinated and everybody still gets sick, but not very sick, the people who are not protected by the vaccine have a better chance of not catching it because the viral loads are lower in people.
I've heard of that.
Yeah. I'm sure you have.
Yes.
At least you're not hard of hurting.
So there are a couple of interesting universal vaccines. One of them, which has been in quite a few trials, it's going into a phase 3 clinical trial. It's called BeyondIvax M001. It has 9 peptides that represent conserved epitopes.
Beyond Vax 001.
Yes, that's right.
It should be 007.
These are epitopes from the M1 protein, nucleoprotein, the head of the hemagglutinin. And they say they're common to 40,000 isolates, and they're produced in E. coli, purified and injected. It's been in 6 clinical trials in Israel and Europe, 2 phase 1, 2, and 4 phase 2. And another phase 2 is coming in the U.S. They've shown safety in a broad antibody and T-cell response. There's currently a phase 3 recruiting with 10,000 people in Eastern Europe. My question, of course, is how broad is this going to be? Even though you have these 9 peptides, is it going to cover just H1s or H1s? I mean, they would like it to cover more, right? They have one that they're testing for H5N1. So that's interesting, but we'll see. We'll see what happens. In 2020, we'll talk about it here on TWiV when the results come out. By the way,
the idea is that they're not just targeting the HA or
it's a mix. Yeah,
it's a mix of those proteins, and they've also tried at least to find conserved epitopes among them. Now, whether those epitopes are exposed enough to target and whether you're going to get antibodies that are neutralizing against it, those are things that have to be sorted out in the clinical trial.
I think it was this one. I was looking at the paper. I think it was this one that said that it worked best in conjunction with the seasonal vaccine, which made me think, about, once again, correlates of immunity, almost as if it works best as a booster, right?
Right.
And not as the primary vaccine. Even though it gives a good, you get a lot of antibodies to it and that kind of stuff. The protection is best if you use it in conjunction with the seasonal vaccine. That, to me, is shouting, we don't understand the immunity.
Right.
Yes.
I was looking at a paper involving this vaccine where they're in a phase 2 looking at protection against H5N1 in people. I don't know how they're going to do that because there's so few infections. Maybe they're going to look at just immune responses. But they say in the paper their peptide vaccine gets around egg allergies because it's not grown in eggs. But it's been known for a while now that it's not a problem if you have egg allergies to get a flu vaccine. So I blogged about that a while ago.
So where does that thing about egg allergies come from?
I don't know.
The dairy industry.
I think it may have just been one of those sort of urban myths that crops up occasionally that because the vaccine is made in eggs, maybe people with egg allergies shouldn't take it. And maybe somebody had an adverse reaction and blamed it on an egg allergy. But if you look at it systematically, it turns out that it's really not causing a problem, even for the very, very rare individuals who have genuine egg allergies.
Okay.
So there's a paper published in 2017, and it was by the Influenza Vaccine and Egg Allergy Practice Parameter Work Group.
They were focused.
So they were very focused. So basically, you don't have to worry about if you have an egg allergy. That we'll put a link to that article.
And not all the vaccines nowadays are made in eggs. There are several that are recombinant vaccines. The one they give to old guys like me is called Flu Block, which is a recombinant vaccine made in baculoviruses and insect cells. So
yeah, I think I got Flu Block. Yeah,
I'm not allergic to flies. As far as I know, so looks okay.
The other universal vaccine is something we have talked about. It's a HA Stalk vaccine being developed at Mount Sinai. The stalk of the HA has highly conserved residues, which are present in many different isolates, and they don't undergo variation, so they're a good target. The problem is the stalk is kind of immunosubdominant. If you put a whole HA into an animal, most of the immune response gets directed against the head, and those epitopes are the ones that vary. Yeah. So Peter Palese and Franz Klammer, no, that's the skier.
Right.
Peter's a skier
Florian
too.
Florian Klammer. Thank you. I'm sorry, but probably Florian would like to ski quickly. They've been working at this over at Mount Sinai.
It's all downhill from now.
The idea is that you can induce a response against the stalk if you immunize with an irrelevant head. So they put first dose, you get a head-stalk combo, and then the second you change the head, and now you direct most of the response against the stalk because you're getting a primary against the head and a memory against the stalk. So,
Alan, I think your wheels are turning trying to come up with a name for that show.
Which one?
No. I was already going to ask if they called their lab the Stalk Institute.
There you go.
That's great.
Or stalking the influenza virus.
So I don't understand this thing about switching the heads. Not that any immunology makes any sense to me, but that one escapes me.
It's basically setting up a situation of original antigenic sin.
Okay.
Where the stalk antigen is the same between each round of the vaccine.
It's like a booster.
And so you can prime a memory response in the secondary and tertiary delivery of the vaccine. And that memory response will clear the antigen faster than a naive response can be made by the naive cells against the head.
But you have to vary the head to get that to happen.
Exactly.
Yeah.
You got to vary the head.
Yeah. Right. Because then the first vaccine, you get this response against the head. There might be something against the stalk, but maybe not detectable. The second vaccine, you've got a totally different head coming in. So the whole immune process has to grind up to speed in order to develop anything against that. But the stalk is the same.
Right.
And so you're boosting that initial response.
That initial small signal against the stalk. And that's the idea behind this. And it's delivered in a way that that I guess the stalk is a little more exposed than it would be in the current vaccine.
So 2 heads are better than none?
Yes.
Or 3. But that's going into a phase 1. And they're going to try both an inactivated and an attenuated version of that. So good luck with that. I know they've been working a long time to try and get that into phase 1. And then the final one has the name Ready Flu. REDEEFLU.
Is that a dessert topping also?
Ready Whip. I wait for Alan to come up with something.
It sounds like a chimney repair product.
I don't want any Ready Flu on my desserts.
No.
Yeah. But if you move one of the E's, then it could be the flu, FLUE. Yeah.
Yes.
So, yeah.
Ready Flu. Yes, that's a good, if you ever start a chimney cleaning outfit, Alan.
Yes.
Ready Flu. So this is being done by Yoshikawa Oka and his colleagues. And this is actually quite interesting. A number of years ago, they made a vaccine, a virus, which is replication defective because it lacks the M2.
That's a great idea.
What's a great idea?
What you just said.
Well, many people have tried it, and we actually have talked about some replication defective vaccines here on TWiV. It's a good idea. I agree. But they've taken out one of the proteins, the genes encoding one of the proteins, M2, the ion channel, and they grow the virus in complementing cell lines that produce the protein so that they can make virus particles. Sure. And these will then enter cells. You need the M2 to enter, but then, and the genes will be expressed, but you won't make new virus particles. And so they are doing this. It's called M2SR is the other name for it because you have to have 2 names that you can't pronounce, right? Ready Flu and M2SR. M2 defective single replication vaccine. So they're going into clinical trials. In fact, there's one over at clinicaltrials.gov that's enrolling safety and immunogenicity in 9 to 17-year-old H3N2. Okay.
So, but this isn't necessarily a universal thing, right? This is just another approach at a live attenuated vaccine. It looked from the description that's here.
Yeah, it doesn't look like a universal. That's correct. Yep. It's just a different approach, right?
Yep.
So the two universals are the peptide and this Stalk Institute.
Stalk, yes.
It seems to me that the notion of original antigenic sin complicates all of this.
Yes.
Right?
Yes.
That was that. If I understand that correctly, it's if you encounter a given influenza strain as your first encounter with flu and mount an immune response to that, then you encounter a different strain later on. There is a tendency of your immune response to answer with boosted immunity that is against the original strain.
Right.
Any shared epitopes, yes.
Mm-hmm.
Yeah.
Right. Mm-hmm. You don't get nothing to the new strain, right? You get something to the new strain, but there's a competition, if you like, with immunity to the original strain that complicates the whole immune response.
Right.
Do I have that right?
Yes.
Mm-hmm.
Yeah.
Yeah, and that's
right.
the general nature of original antigenic sin, as we call it, is that your immune system kind of is biased toward the things that it's encountered before.
Yes.
So whatever your first, I guess you could say you never forget the first time you flew. So whatever your first exposure is, then that's going to shape your responses to all subsequent flu infections and flu vaccines.
Sure. You see these maps from the CDC and other health agencies as well, showing the intensity of influenza for a particular year or a particular season. And what I'm wondering is, after that season is through and there were lots of exposures and lots of infection, is that enough to prevent that from showing up on next year's map? And the answer is, do we know how many people are actually exposed in a big epidemic localized, let's say in the state of Rhode Island or New Jersey? Do we actually know how many people who didn't get sick were still exposed to the virus? That's what I'm saying. Without vaccination or anything else, is there just a low infection rate of the virus, or is it something else that prevents it from going on and reproducing?
Well, people have done serological surveys which would tell you that, right?
Yeah. So what does that show?
I don't remember those specific data, but I know there is some data on people who were born before and after 1968, when there was a switch from group 1 viruses to group 2, or it could have been vice versa, and differences in their susceptibility, it looks like, to H7 versus H5 avian flu viruses, because, again, those are in the two different groups. And so if you were born in a time where your first flu exposure was against a group 1 virus, then you are particularly susceptible to group 2 viruses. So people born before 1968 seem to be somewhat protected from H5N1 and vulnerable to H7N9, and those born after 1968 have the reverse.
We'll have to do an episode on this at some point, because this whole idea of the first infection being really important. Yeah, there's a lot of data on it, and your question is valid. I don't know the answer.
Mm-hmm.
We did an episode on, the only reason I know anything about it, original antigenic sin, is we did an episode years ago, but it needs to be refreshed.
Yeah, I think so.
We'll revisit.
Yep.
But our memories of that first episode will shape how we cover it the next.
That's right.
Definitely.
For sure. And there are other ways that you can try to prevent influenza. As Kathy has noted, there's some interesting new antibody stories that have just been published. I guess these are single chain antibodies, right, Kathy?
They're from llamas, which like camels have these single chain antibodies.
Exactly.
And so it's an interesting approach that involves delivering the important material by way of an adeno-associated virus, a Parvo virus. So
cool. I want to do that, and then we'll see how Alan makes a title for that, with the llamas.
So the other thing I wondered is that when you hear about these large epidemics of influenza, and then you see movies or newsreels, by the way, of, let's say, in Asia, they take you to China. Well, newsreel, sure, that's old school. And then they show you. Are we
taping this episode?
Got the real.
No, I mean, that's right. Not someone there transcribing it manually. I'm curious as to whether or not a face mask. You see Tokyo, for instance, everybody's got a face mask on. The moment they hear about this influenza outbreak, they're going to protect themselves from this infection. Does that actually work?
We've talked about that a little bit. The conclusion is kind of, sort of, if it's worn properly.
Right.
But it usually isn't.
If you wear it every day, you're not going to wear it
different
properly.
kinds of face masks. Some are useless in preventing.
Yeah, I know. I agree with
so
that.
forth.
I agree with that.
And people don't wear them consistently, and they don't wear them properly or fit them properly. And it's got to be, you really need to have the proper, at least an N95 respirator, I think. And and there's still some ongoing work on that, mostly for hospital environments. But I don't think there's any evidence that the general public's application of that has had much impact.
Right, and yet they still wear them.
Of course, because it becomes,
it's fashionable.
Yes, in fact, and there are multiple reasons, it turns out, why particularly women in Japan wear those face masks as a fashion accessory. And it apparently it reduces the rate at which they get harassed on the train.
How interesting.
Wow.
So
yeah.
Steve Morse, a long time ago, like before episode 90, came on and talked about face masks.
Oh.
And the name of the episode was Arsenic and Face Masks.
I missed it.
Yeah, I know.
I wasn't there.
You weren't there for a lot of them.
I know.
All right, let's move on to a paper. This was sent in by Sommer, who wrote, "Hello, I would love to hear this paper covered if it's of interest to you. And if it's not, he or she would not like to hear it covered."
Apparently, it does have interest.
But it is of great interest, Sommer.
Here we go.
And
yes.
It is... I saw this a couple of weeks ago and caught my eye, and thank you for bringing it to our attention. An atypical parvovirus drives chronic tubulointerstitial nephropathy and kidney fibrosis. It's published in Cell, and it comes from a big consortium: University of Sydney, University of Technology in Sydney, Memorial Sloan Kettering Cancer Center here in New York, Johns Hopkins University School of Medicine, the Military Institute of Hygiene and Epidemiology, which is in Poland, Monash University, Tufts University School of Medicine, the Australian Genomics Research Facility, University of Melbourne. I had to say that, Melbourne, because I learned that. Royal Prince Alfred Hospital and the Medical University of Vienna, and finally Genentech, San Francisco. Wow.
Yeah.
And there are tons of authors.
The first author is Ben Rediger, and the final author is Wolfgang Wenning.
I thought you were going to say Ben Rediger because it was a completed circle.
No. Rediger and Wenninger. But I was double checking to see if there were two first authors, but there's just one.
By the way, the name Sommer is an Arabic male name.
Thank you.
Okay.
Meaning person who chats nicely at night.
Oh.
Wow.
Only at night.
A late night host.
Not during the day.
Well, probably chats nicely during the day, too, but especially nicely at night.
All right. So this is a cool story about a
it's never the summer during
disease
the day,
that has been happening to laboratory mice for over 40 years, and no one could figure out what was the problem, what was causing it. And this is very interesting. Wow. So it's a chronic kidney disease, and it affects middle-aged mice.
Immunocompromised.
Especially immunocompromised.
And many animal facilities have immunocompromised mice, knockouts of various sorts that are used to do experiments, and those are the ones that preferentially get targeted. And this has been happening in the United States and Australia. And that's why you have people from the U.S. and Australia on this paper who have looked at this. So this starts back in 2010 when at the Centenary Institute Animal Facility in Sydney, they noticed unexpected deaths in middle-aged mice. That statement is so funny. Middle-aged mice.
Middle-aged mice.
Right? Young mice and old mice. Middle-aged mice. And it looked like it was kidney failure. The kidneys were shrunken and pale and pitted and highly fibrotic when you sliced them and looked at them under the microscope. And all the other tissues seemed to be okay. And this, as Rich said, mostly in immunocompromised mice like RAG1 null mice, they don't have TB and NK cells. Is that right?
Right. That's right.
Mostly T and B.
And NK are okay?
Yeah, because NKs don't need to do VDJ recombination.
Then there's skid mice, which are also messed up. And this happens.
They're missing NK.
They're missing NK as well as T and B, right?
Yeah.
Okay.
And nude mice, which are not quite as messed up, right?
They're just missing T cells because they don't have a thymus.
Right. So these die between 200 and 300 days. I guess that's the middle-aged mice, yeah?
I guess so.
So that less than a year.
Yes.
The more detail in what's going on in the kidney, tubular degeneration, the tubular epithelial cells degenerate, necrosis, and you have enlarged nuclei with inclusion bodies in the nucleus.
Dun, dun, dun.
And if you're a DNA virologist, immediately you're thinking, that's my
cue.
But they didn't see any viruses. They did EM and they didn't see any viruses in there. But when they stained the tissues with acridine orange, it binds to single-stranded nucleic acids, they saw some staining. And so they decided to sequence these mouse kidneys. They took out RNA from the kidneys, they sequenced it, and they found a lot of unassigned sequences and put them together and ended up identifying a parvovirus, a novel parvovirus. So they got a complete 4,442 nucleotide viral genome, which they then went back and did other sequencing to confirm it was really there in the kidney, and they call it mouse kidney parvovirus, MKPV, which is the subject of the rest of the paper. Yes?
What I like about it is that they specifically did RNA sequencing because even though it's in the nucleus, they didn't want to be biased, and so they wanted to cover their bases in terms of RNA and DNA viruses.
So the RNA would pick up an RNA virus, and of course DNA viruses also make RNA transcripts, so it would pick those up as well.
So parvoviruses are eeny-weeny guys. Single-stranded DNA, just 4 kilobases in length. Naked capsid virus, just 2 genes. A capsid protein and a non-structural protein that basically is required for replication. The most familiar human parvovirus is B19 that causes a disease called fifth disease that most people are familiar with. It's commonly called slap cheek syndrome. Okay, it causes a skin rash that looks like looks like your kid's been hit in the face, right?
And parvovirus really means small virus.
From the Latin word parvus, which means small.
Yes. Parvus.
This week in vocabulary.
Parvus would be a nice name, except you'd probably be insecure your whole life. Right?
Parvus, would you mind refilling this drink for me?
I think I've heard Parvus as a name. It's interesting. And of course, there are parvoviruses of cats and dogs, and that you have to immunize your pets because they're very bad diseases.
They are.
There are also parvoviruses of mice that are
minute virus of mouse, mini mouse.
It doesn't get much smaller than that.
Those have been model systems over the years. Yep. So they've made some primers and did PCR, and they look in various parts of the mice, and they find the virus, or they find viral DNA in mouse colonies where there has been kidney disease. But where there's no kidney disease, they don't see it. They find the virus in serum. They also find it in the feces, although they say there's no apparent pathology in the GI tract. And then they do some co-housing experiments.
I love these experiments. These are so cool.
So they have infected mice, which are PCR positive. That's how they assess them. And they co-house them with, in this case, they were both RAG1 knockout mice, and these were obtained from a virus-free vendor.
Huh.
Although, if you don't know about the virus before, I don't know. I guess in general, virus-free.
Right.
Yes.
They're only virus-free for whatever viruses they can assay for.
Yeah.
Right.
I guess they checked them before they did this experiment, right?
Yeah.
So they put them together, they put them in the same cage, and mice tend to exchange things. And after 50 to 80 days, viral DNA could be found in the serum and urine of the co-housed animals. And they got sick, they lost weight, and they died of kidney disease.
And somewhere in this paper, they even did an experiment where they took bedding from infected mice and put uninfected mice in that bedding without the infected mice, and the uninfected mice caught it. And they conclude from that there's an oral fecal transmission of this thing.
Mm-hmm. It could also be a urinary transmission because it is in the kidneys.
Yes.
Co-housing.
Yes.
Experiments. And you mentioned that it's also in the urine, which makes sense, right? Because the virus is most likely replicating in the kidney.
Right.
And I'm not sure. They say at this point, and I guess at this point they haven't done any experiments, but they say, this is this virus is controlled by the adaptive immune system, which is missing in these knockout mice that we've looked at, because mostly it's those mice that get sick.
Right.
So this has been all done in Australia, and then they did similar things at Sloan Kettering. And
there's this remarkable pivot in the paper, actually, as you're reading along. The mice at the Centenary Institute had this and these histological features. They were identical to those associated with morbidity and mortality in mice at Memorial Sloan Kettering Cancer Center.
Yeah, right.
Like it's right next door.
Quite a distance away, actually.
And I'm envisioning, I don't know if this is how this actually happened, but I'm envisioning maybe somebody giving a talk at a meeting. And then someone comes up and says, we have the exact problem with our mice. I could imagine something like that happening. And then, of course, we're talking about the mice at Sloan Kettering, and they go and look and they find the same viruses in those mice.
Yep. So they have mice with the same kidney disease, and they found the virus in them and found it in urine, and they do some co-housing and same thing. So it's the same virus in both places, interestingly. They do some other cool. Actually, this is a lovely paper. They do a lot of very nice stuff.
This, yeah.
And we were writing a grant where I said, "Look, this is exactly the way we should put it, because as you'll see, But here they look at in situ hybridization. They can see that the viral sequences are just in the cells of the kidney that are being affected. The ones with the inclusion bodies, those have viral sequences in them, which is really cool. They also make antisera to a viral protein and stain and see viral antigens. And then,
I love the way they make the antiserum. They co-house infected mice with wild-type mice who don't get sick, presumably because they make an immune response. And then they take serum from those wild-type mice and show that reacts with recombinant protein from
nature's way.
From the
Very
virus.
clever.
That's unbelievable.
I could see the day when they said, hey, we could get serum from these mice and do a staining.
Exactly.
And they can see the viral antigens.
Or how come these guys aren't getting sick?
But get this now. Listen to this part. They did mass spec of the bloody kidneys to identify viral proteins in them.
Wow.
I mean, that's amazing. I wouldn't do that.
Well, they didn't want to hear Vincent complaining about how DNA is not virus.
They didn't do a plaque assay.
Well, they couldn't.
Well,
yeah.
Indeed.
That's a problem. These viruses are notoriously difficult to grow.
We'll get to that, but
we'll get to
that. So they did this this liquid chromatography tandem mass spec LC-MS-MS, and essentially looked at all the proteins in the kidneys and picked out the viral peptide.
All the proteins.
This is an incredibly powerful technique.
It's amazing. It's amazing.
You can do this now the way that technology is. And so they found, sure enough, you've got viral proteins in these infected kidneys.
This will come back later. There's more use of these data later on.
How fast are these machines, by the way?
I think they're very fast.
Very fast.
Yeah, they're fast. This is an experiment you do in days. This is not years. You break up the proteins to peptides, and then you can match that against the database. And that's the key to this also, is having a database of peptides that you say, this is from this protein, and so forth.
This is a mass spec is a technology that's had a development that rivals sequencing. Where it used to be really difficult and really expensive, and you couldn't use complex samples, and now you shove a kidney into the
machine. survey an entire country like that.
The other point I want to mention is that they compare the viral loads in immunocompromised versus immunosufficient mice, because as Rich said, they get immunosufficient mice get infected, but they don't seem to get disease.
I hadn't heard that term before. I thought immunocompetent was the usual one. I saw immunosufficient.
I heard that or saw it recently. Yeah, I kind of like it.
Yeah.
So anyway, there's 10 times more viral DNA in the immunocompromised mice. So presumably more virus and more severe pathology. Because the immunosufficient mice do get infected, but they don't seem to develop a disease as frequently. I mean, they say here some of the histopathological features have been reported in immunosufficient mouse strains. So it can happen, but I think it's much less frequent.
And that's why they make that claim that this virus is controlled by the adaptive immune system.
Yeah.
Right.
So what is this virus? They've got the whole sequence. You can go and compare it to everything that's out there. It's a really diverged parvovirus. Not very similar to all the common ones, but mostly similar to viruses from old world fruit bats and common vampire bats.
Common? I would call that uncommon no matter what. But
so parvovirus from those bats is very similar to this. And they say it's probably a new member of a new genus which has been unclassified. So it's very divergent. They do phylogenetic trees and so forth to show that. It's also smaller, 25% smaller than typical Parvo Viruses, and similar to Desnovirinae. And let me ask the crew, does anyone remember a TWiV on Desnovirinae?
That sounds familiar.
Isn't this a typo and they mean denso?
Yeah, maybe it's denso.
Denso, yeah.
But we did a TWiV on that one. Do you remember what it was? Let me do a search. Densovirinae. It was the starfish virus.
Oh.
Densovirinae.
I remember the starfish guy. That was great.
What episode number?
I didn't get the episode.
You know, you can't look these things up because Alan. I was telling Harper the other day about,
must be something in the water.
That's it. Sea star melting disease. So I can't believe they have a typo, Kathy. That's horrible.
TWiV 315. Yeah, I saw that and I thought, what? And then over in the figures, they have it as denso. And later they talk about it as denso.
Gee.
Yeah.
They do some other experiments where they look at the course of disease. So the way they infect mice is they co-house them.
Right.
Because they haven't got virus, although they should. We'll talk about that in a bit. And, you know, when you co-house them about 100 days later, they're all seropositive or virus positive, PCR positive. And then the blood urea nitrogen goes up, BUN, and that's kidney dysfunction, right? That's a sign of that. And then between 150 and 250 days, proteinuria goes down, protein in the urine, so kidney dysfunction, weight loss, and then they die. And this is, again, the virus is in the kidney, but it's in serum. They don't find it in every organ. And each kidney contains 10 to the 12th to 10 to the 14th genome copies. It's a big number.
That's a pretty good load.
I think it is.
Yeah, it's a lot.
I'm surprised they don't see particles.
Yeah, I am too. They should, right? The last part is really cool. This is where...
Well, they are small.
This is where pathogenesis, here you go, of MKPV-induced kidney disease. They do transcriptome analysis, and they also go back to their mass spec data, and they say what proteins are going up and down. And it's really interesting because they see going up established kidney injury biomarkers, which makes sense. So they're proteins that we know of in urine, for example, that correlate with kidney disease. They're going up, which makes sense. They see an upregulation of like transforming growth factor, collagen, the fibrotic tissue you see between the cells, which is characteristic of the disease. That's putting collagen down and fibronectin, and they see all of those go up. Connective tissue.
Any heat shock genes?
Growth factor. I don't remember seeing that. Immune system genes upregulated, of course. They see an expansion of macrophages in the kidney, and they get activated, which they can tell by genes that are going up. Complement genes are also turned on in the kidney. And here they went back to their mass spec and looked at proteins that go down and that go up. And these are reflective of kidney damage, basically. It's really quite interesting. They also break up the kidney and do flow cytometry and say what's going on in them, what kind of immune cells and other cells are infiltrating into them. So their conclusion is that the infection drives a loss of the epithelial cells, an expansion of activated macrophages, and development of myofibroblasts within the kidney. And so one of the things that I think is really interesting is that, they're trying to position this as a model for human kidney diseases, which are not easy to study. They say in mice before, to get this kind of tubulointerstitial fibrosis, you have to clamp off the ureter, and that backs up urine into the mouse kidney, and you get a similar disease.
Hydronephrosis.
And so they say some of the markers of that disease are similar to what we see here. And here we can put a virus in, so that may be a good model of that. And then finally, in people with kidney disease, it's hard to know what's going on in the kidney just by looking in the urine, but they're saying maybe we can have some biomarkers that
early event
useful. And they find some that are common between the two diseases, this virus disease in mice and human kidney disease. So maybe some other biomarkers they find in mice could be validated in people as well. So that's cool. So then, so if you notice, that's the end of the experiment. And they say,
does this virus cause this disease?
Does the virus cause the disease? They actually say our findings would be strengthened by inoculation experiments with pure virus, which you are presently unable to propagate in vitro. So I guess they've tried a lot of things. They've probably tried
organoids,
taking extracts of these mice, kidneys or whatever, putting them into organoids or
it would have been nice if they had told us if they had done some of those things. Like if you just made a homogenate from the kidney and and put that. I mean, a lot of viruses in the olden days, when there wasn't good cell culture, would be passaged, mouse to mouse. So,
I mean, especially if someone else wants to work on this, they would see, they've done this already, right? So we're not going to spend 6 months doing it. And what Rich wrote in the notes, tell us what
parvovirus DNA is infectious.
Yeah. So
that would be been my first thing, is to try and infect with a clone DNA molecule. They must have tried.
I'm sure they did. Yeah. I'm sure they tried.
So I'm forgetting right now, but only some parvoviruses are dependoviruses and require another virus for their replication, right? Because that could be one idea. Does this virus depend on something else, and that's why they can't propagate it?
Yeah, like a helper virus or something.
Right.
Yeah. I mean, the other parvos, these bad viruses, I'm sure they don't have replicating virus, so we don't know. And maybe it's too distant from the autonomous parvos, like the minute virus of mice, to be able to determine whether it would need a helper or not. So does anyone know, in the helper-dependent parvos, is there something missing, or it's the same gene complement as other parvos?
I believe there's something missing that either adeno or herpes can provide, at least in the human AAVs.
Let's look at viral.
My impression has usually been that what's really required is for the cells to be stimulated to want to do DNA replication. So there are some, B19 is homes to erythropoietic cells, because they're rapidly proliferating. And the effect of adeno and herpes is to the genes involved, I think primarily, is to stimulate the cells to at least pretend that they're going to replicate.
You need E1, E2, E4, and VA from adeno to be as helper functions. So the E1 would be getting the cells to replicate, I guess, right?
And you need VA?
Yeah, because when you make AAV vectors, you throw that in on a plasmid.
Yeah, our lab started making some AAV this summer, and it was much more complicated to make than I thought because of the needs for all of those.
Yeah.
But the VA to counteract PKR. Okay.
Yeah.
Just the surprise.
Our listeners out there must think we're just practicing reciting the alphabet.
Well, we're practicing virologists.
I mean, the E stands for early protein, yes?
Yes.
And VA stands for virus associated RNA.
Thank you.
It's a small RNA that.
And what does the PRV stand for?
PKR.
PKR.
IF2 alpha kinase.
PRV is pseudorabies virus.
There you go.
That's a herpes virus.
This is like, I love this.
But it doesn't cause rabies.
Okay, for 20 points.
Any more confusion? Why don't we have an episode where we just say nonsense for an hour?
I thought we've been doing that
that's
already.
What people think.
Yeah, exactly.
So the reason this virus comes up is because, Koch's postulate
Koch.
Pronouncing that will cause something to come up.
says one of the things you have to do to prove that a causative agent of disease, you have to recover the agent, grow it in culture, put it back in the animal, show it causes disease, and then get it back out. And of course, they haven't done all of that here. But this is an interesting issue because now we think probably Koch's postulates, we can't do them all the time because you get a lot of viruses in silico, and so you can't grow them.
It was predicated on Vibrio.
And so there's actually Koch's postulates for the 21st century where you don't have to do all the things that he did, but you do other things. And so they say our data fulfill most of the new criteria. So they say the virus is
these
present. These
criteria are outlined in a paper by Fredericks and Relman that has been widely cited for this.
Present in all mice with inclusion body nephritis, but not in healthy controls. And it's found preferentially in the kidneys. Second, the virus viremia usually precedes disease, and the copy numbers correlate with disease severity. That's cool. Third, the parvos are known to assemble within the nuclei, and they see inclusion bodies, right? And fourth, in situ reveals viral sequences in affected but not normal tubular cells, even within the same kidney, right? You see infected are causing the pathology. So the only thing that could be going on here is there's something else that's going along with MKV, and you're not seeing it, right?
I can just hear the lawyer for the other side saying,
yes,
Your Honor, these are all circumstantial evidences here, and how can we possibly convict this poor virus on those grounds?
So based on what we see here, if this were people, would you make a vaccine against this agent?
I think it'd be worth trying. For me, a really, really telling thing is that this virus is in all the mice they looked at that had this condition. Mm-hmm. You don't find a mouse with this type of kidney failure that's not got this virus. That's pretty incriminating.
I'd say.
I think so.
Plus the other...
the inclusion bodies, it's there with the viremia. You've got this, it's the viremia is in the kidney where the disease is taking place, and it all fits the pattern for parvoviruses. So it's really, it looks pretty solid for this.
Mm-hmm. I also think it's of relevance for mouse facilities now. They have to start looking for this virus, right? They look for a bunch of others.
Sendai, it used to be when I was doing stuff, Sendai. But I wanted to ask, can you take from the cohabited normal immune competent or immune acceptable mice, could you take immune sufficient, and during the course of the growth of the animals that don't have that advantage, could you take their serum from the immune competent mice and protect the others from old age syndrome?
Yeah, I would say,
and that would be absolutely spectacular.
Yeah.
Passive immunity, yeah.
Mm-hmm.
So then your vaccine has great hope.
That would be, well, if this were humans, that would be the type of experiment you'd want to do in an animal model to try and
that's right.
Yeah.
That's right.
Figure out if that was a good correlate.
Did we say what the conclusion actually was?
Well, they believe this virus is the agent of this kidney disease.
No, but what is the actual composition of the inclusion?
Well, there are viral sequences in there, so presumably
it's just a bunch of DNA clumped together like that.
Well,
it looks like an
aren't these assembly sites for these viruses, right?
Probably.
in the nucleus.
Replication sites.
Replication.
Sites.
So clearly the DNA replicates there, and it probably assembles virus particles.
But when you stain it, I looked at the sections. They just looked like big round blobs.
Right.
So there's no protein from the virus in there?
I'm sure there is. Capsid
there is?
protein.
Okay.
Yeah.
Okay.
Sure.
Yeah, the serology stains capsid proteins.
And also there's a rep protein that helps DNA synthesis. It's going to be in there as well.
Very cool.
It might be interesting to see if it's possible to purify enough of this stuff from mouse urine.
Mm-hmm.
You know, if you can purify a virus from mouse urine, and then
what would you do with it?
Then try and infect mice with it.
Yeah, sure. I bet they've done it.
I mean, it's not gonna be definitive because you'd have to. Well, I guess you could, if you can get antibodies against the virus, you could purify it on an antibody column, so you're not getting other viruses.
Absolutely.
And then try and do. This is not an easy experiment, and I'm not saying they should have done it for this paper, but it would be.
But if they have done it, it should work, right?
Yes,
because it's the same virus you purified out.
Right,
I mean, trying to grow it in culture is another issue. I could see why that doesn't work.
But that would come very close to fulfilling Koch's postulates, no matter how you say his name.
Purify the agent.
Yeah, for sure.
Yes.
But yeah, I mean, either way, that would be easier than a cell culture system. Yeah, I agree with that.
If I'm not mistaken, Koch's postulates were formulated before we even really knew what viruses were.
They were.
Yes.
They were.
And so you can't even satisfy Koch's postulates really formally with any virus because you can't culture it in the absence of cells.
Correct.
Mm-hmm.
Right.
Now, if you have a colony that's got this virus, you've got issues.
You got it.
Because they say the only way you can get rid of it is to rederive the mice, which means from embryos. And so that's hard. Or you can just take all the mice that are infected and get rid of them. But then you got to get new ones anyway.
But if all your rag knockout mice are infected, you'd
you'd
have
to
you got to nuke the whole colony.
Well, you can buy them. You can buy them. So I guess if you have something,
but they might
still have
be infected from the supplier.
Yeah,
That's
they could.
Thing.
Yeah, if they are infected, you could check that.
If you rederived them, you'd also presumably clear them of other viruses. So I know that Skip Virgin had all the mice at WashU rederived so that they didn't have mouse norovirus.
So tell us what you do when you rederive a mouse to get rid of this infection.
You deliver pups by cesarean section, sterilely.
In an isolator.
So you go back to embryos.
And then how do you raise those pups with a pseudo-pregnant mother? Because you don't want to leave it with the original mother, right?
Feed them.
Yeah, I don't know what you do once you've delivered them, actually. I hadn't thought about that.
You can hand feed them a liquid diet until they reach adulthood. And I know someone who actually did that at Notre Dame during the germ-free days when I was there. So
this is not a trivial process.
Not a trivial.
No.
But once you've got adults, then you can breed.
Then
you're all set.
The last question I had was, they didn't look in wild mice. And I would like to know in the wild if it's out there.
Yeah, good point.
Right? Because maybe, if it makes immunocompromised mice sick, then maybe it's out there in the wild and they're fine, right? Mm-hmm. And how do they get from there to the facilities? Well, that's actually not a big deal. That happens all the time. We have our mouse facility got contaminated years ago by, if you walk in off the street, you will have mouse viruses on your feet. And unless you wear things over your shoes in the mouse facility, you will track those viruses, and eventually they'll get into the cages and infect the mice. So that's not a mystery how the virus would go from outside to inside, unless you have really good barrier facility.
Right.
A lot higher than just your standard SPF.
Yeah, it has to be really good. And here they're actually rebuilding the mouse facility. Spending many millions of dollars because it turned out there were wild mice in the walls of the building that were getting in to the facility at night, and they get in, they eat the food, they defecate in the cages, and of course they leave their viruses behind.
Oh my gosh.
Isn't that horrible? What did we say last week? We complained about Columbia's Wi-Fi. Today
facility is infested with mice. Well, isn't that no? You
don't get it. That's
not just the ones we want.
That's right. Wild type. Yeah. Well, some of them are kind of crazy, but you know.
do a couple of emails.
Lordy.
Our first one is from Mark, who writes, Dear TWiViDae, here in Northern California, the weather is sunny with clear blue skies, temperature in the mid to high 70s, or about 24 to 25 for those who measure in Celsius. In other words, classic, beautiful California autumn weather. Last Sunday, I went to the local firehouse for a free flu shot. My nomination for listener pick is a page on the CDC's website. Features an animation showing spread of flu throughout the U.S., plus D.C., Guam, Puerto Rico, U.S. Virgin Islands as the flu season progresses. It has a small database allowing you to see flu progression by season going back to 2003. Clearly, some years are way worse than others. On TWiV, it has been noted that flu transmits more effectively in colder weather. Questions. Is there an optimal temperature range for flu transmission? Have any studies explored the probability of transmission based on temperature? I got my flu shot. Did you, Mark? Yes. In fact, we talked about it earlier.
Yep.
A long time ago on TWiV, we talked about the Palazzi lab, and there are others as well who have worked on this, showing that low temperature and low humidity are important for flu transmission, at least in a guinea pig model. So here in the winter, low temperature, low humidity, the idea being the particles can float further in the air with less humidity. But it's not so simple because the tropics also have flu, and it's not low humidity and low temperature there. So I think we don't know all the answers.
I think it may, in the tropics, I think it's more of a wet season, dry season thing, but it's still not going to be as low humidity as, say, New York in the winter.
So I did paste in one review, and the final line that I pasted out of the abstract is the mechanism of virus survival in the cool, dry, or humid, rainy conditions is largely determined by the presence of salts and proteins in the respiratory droplets. Wow. But, somewhere else, I think earlier in the abstract part I didn't paste in is just kind of saying that we still don't have a totally good handle on it.
No.
But people do study it.
Yeah, for sure.
You bet.
Dickson, can you take the next one?
Izzy writes, I noticed on TWiV 516 that Jeremy Lubin took special care to credit each lab that came up with an advance by name. This may happen in every TWiV, but is this the first time I've noticed that? It's so important to give labs, even your competitors, especially your competitors, credit for their work. Competition and collegiality are not mutually exclusive, and it's important to remember we all want the same thing. I hope every guest is as diligent as Dr. Lubin was in giving credit where credit is due.
He's very good at that.
Yes, he is.
He tells stories and includes everyone. Yeah,
it is important.
That's right.
I totally agree. Right. Brianne, can you take... Sorry, Alan, go ahead.
No, that's all right. Go ahead.
We'll get to you.
Yeah.
Brianne.
Sure. Andy writes, the GG weather. Please edit the beginning of the podcast to reduce the casted small talk to under 30 seconds. I hate to be so blunt, but in episode 517, I skipped into 6 minutes and 18 seconds before I started listening. The content is very interesting and provides a great public service, but all of this could be lost if your listener base drives into a bridge abutment while trying to skip to the topic. The number 30 seconds is inspired by other podcasts with ads. Ads over 30 seconds I skip over. This comment applies to all of the Twix podcasts. Again, I really appreciate the information and discussion. I am an aerospace engineer, and it is not easy for me to access biological research or spend the time to understand it. Thank you. Andy from Rochester, New York.
Well, Andy, you're not the first person to write in with this complaint, and we keep doing what we do.
We have a lot of public service stuff in the beginning we'd like to get through. A lot of people ask us for help in announcing meetings and courses and so forth. Yep. And I try not to make it too long. But
that takes up more time than the weather, and no one objects to that.
What do they say? Don't complain about the weather. You can't do anything about it.
Yeah, exactly right. If you don't like it,
actually, Mark Twain said everybody complains about the weather, but nobody does anything about it.
Well, he also said
turns out we have been doing something about it.
If you don't like the weather, if you don't like the weather and you live in Connecticut, just wait a minute.
So, your model for this initially, Vincent, was we have all these conversations in our offices and we ought to make them public.
That's right.
Okay?
That's right.
And all of that kind of stuff about the weather and everything else is part of those conversations.
You got it.
I come in the morning. I say, Dickson, look at what's doing outside.
I know. It says, which way's the tide going, Vincent? We have a buoy out in the middle of the Hudson.
You're absolutely right. Well, what always I come back to is that almost every person who writes in tells us what the weather is where they are.
This guy from California, by the way.
Andy didn't.
Andy didn't know.
I'm very curious about the one from California, though, because that must have been an old email because it's really not so good right now.
Yeah, it was a couple weeks ago.
Yeah, I could probably guess about the weather in Rochester at this time of year.
Yes.
Alan, can you take the next one?
Sure. Eric writes, I've been subscribed and listening for years. This is the first feedback I felt I wanted to weigh in with. I wholeheartedly support you all inviting Jens back as you seem to intend. I loved the episode, number 508. You all have a great chemistry, and he complimented it well, in addition to being very interesting and evoking a lot of laughter from me. In Sedona, Arizona, it is sunny, clear, and a comfy 72 Fahrenheit. In addition, we felt a small earthquake yesterday, which was the last one reported in 2 years.
Wow.
Thank you, Eric.
Sedona is a wonderful place.
It is great.
Yeah.
Really cool.
They make a lot of Western movies there.
Yeah. All right. Has anyone not read an email?
I read one of the...
Oh, wait, why don't you take the last one, Rich?
Connor. Has Kathy, have you read an email?
I did in the beginning.
Okay. Mm-hmm. Connor writes, hi all, from a beautiful Dretch morning in Scotland. Last year, I sent in information showcasing the virology coloring book that we at the MRC University of Glasgow Center for Virus Research produced as part of that year's MRC Festival of Medical Research. This year, we have helped produce another tool to help engage people with the world of viruses, and we thought that you and your listeners would like to hear about it. This time we, which is Ed Hutchison, an MRC fellow here, and I, Connor Bamford, teamed up with the acclaimed Glasgow School of Art and an extremely talented master's student, Rachel Suett, S-U-E-T-T, to make a free augmented reality app for your Android phone. Sorry, Apple supporters. In the app and accompanying poster set, you can explore the structures of 3 important viruses: herpes simplex viruses, influenza A viruses, and Zika virus. There is even a quiz to test the knowledge that you have hopefully gained. You can find more info over our CVR blog, and he gives a link to that. I include specifics to download the app at the bottom of this message. So please have a look, download the app, and play. Then let us know what you think. All the best. Keep up the excellent work, Connor and Ed. And then he's got instructions for downloading the app, which you can read in the email from the episode.
Right.
Well, I don't have an Android phone, so I can't try it.
Right. I'm in the same boat.
I'm working on downloading now.
You have an Android phone?
I have an Android phone.
All right, so play it this week and let us know how it is.
Well,
in his blog, he's got some images of what this looks like.
Right, let's do some picks. Brianne, what do you have for us?
I have an article that I read yesterday, and I was really surprised by this process. I'm not entirely sure what to make of it, in fact. But Nebula Genomics is a new startup founded by George Church, who does quite a bit of work up at Harvard in genetics, that is offering free genome sequencing. So usually people have been doing things like 23andMe, which looks at SNPs, and this is actually a full genome sequence for people. They're using special technology, blockchain technology, to keep the data private. And the idea is that you can choose to share your data with researchers doing different types of research and get credits to either get higher quality sequence or potentially money, although they haven't figured that out yet. And so it's just this idea of getting a full genome sequence and controlling your information. And like I said, I hadn't quite thought about it before and had no idea what to make of it entirely. So I thought that it was something people should think a little bit more about.
That's cool. I like that.
And what could possibly go wrong?
Yes, what could possibly go wrong?
Exactly.
So, and you can opt to pay $99, which is about half price compared to 23andMe. But for $99, you get 0.4x coverage at first, whereas with the free test, you get 30x coverage, and 30x is a measure of really high sequence accuracy.
Right, and I think that if you do the 0.4x coverage and then decide to share it, you can get credits to get the 30x as well.
Cool. I'm going to do it.
You might want to check with your kids first.
Really?
I don't know.
Yeah, I understand.
I really like the data I have from 23andMe.
I'm going to ask them this Thanksgiving. They'll be all there. I said, do you mind if I have my genome sequence and make it public? And they'll say, oh, no, and then I'll do it.
I'm going to wait another. I'm going to wait another 5 years when Medicare will pay for it.
Yeah,
they'll post it on in your Facebook.
Yeah, exactly.
Alan, what do you have?
I have. Oh gosh, what do I have? Oh yes. So this is a paper that I saw just recently, and it's on PLOS One, so everybody can access it. It's multi-institutional study of GRE scores as predictors of STEM PhD degree completion. And it turns out that GRE scores are pretty useless in predicting who's going to finish their PhD.
I think we had data on this before, but
this is not the first time somebody has pointed to the GRE and pointed out that it's kind of BS. But this is yet another brick in that wall.
So let's get rid of it.
I would. Sure.
Yeah. And I'm saying that as somebody who's done well on standardized tests his whole life and probably benefited tremendously from that, but this is really just not doing anything useful for anybody.
Unfortunately, ETS will object, right?
Of course.
Yeah, sure.
The educational testing service that
you get rid of it, you knock out a whole industry.
Well, yeah.
So,
but it's an industry that's not serving any good.
Can I ask, what do the MCATs predict?
Probably the same thing.
Probably the same.
No. That's not true.
What do they predict, Dickson?
They predict that you will finish medicals.
Really?
Yeah.
Do they?
They do.
Yes, they do.
Because I hate those even worse, and I never took them, but...
No, I don't hate them. I'm still on our admissions committee. I use them, but I don't go by them entirely, of course.
Now, why should there be a difference?
Is there a big problem with people finishing medical school?
No, when I ask another question, why don't these people finish their graduate degrees? And it's not because they're not smart. It's not because they don't know what they're doing. Maybe. I don't know. The people who do the best on the GREs go off and form their own companies before they graduate.
Maybe.
It could be. I mean, something...
But this also dovetails with data we've been getting on SATs, which have pretty much been shown to be, well, I'm not going to say a scam, but not terribly useful in predicting academic success at the college level.
How do you? Even if I agree with that, and I'm not sure I do or not because I don't have any data of that. But okay, I'm on the admissions committee for Harvard or for Columbia University.
The top predictor of success in college is good grades in high school.
Good grades, okay, fine.
Yes.
Well, we look at those too, by the way,
by a huge margin.
Yeah.
And what the test scores have been doing is essentially
it's one bad day.
Essentially catapulting lesser students into better colleges, and the students that they're catapulting in are overwhelmingly folks who look like me.
Well, there's a oh damn, what the hell's the name of that Newsweek? What the hell's the name of that business publication that comes out with all the evaluations of the colleges?
U.S. News and World Report.
That's the one. You have no idea of how sensitive deans of medical schools and graduate schools are to those scores.
Oh, yeah.
Or undergraduate.
And they think, and they're wrong, I think they're wrong, that students actually read those magazines and they say, oh, I think I'll go to this school because it has a higher SAT or a higher MCAT or a higher LL. That's crazy. They don't do that. They just don't. They go to a place because it resonates with their personality. That's why they go. And it drives everybody crazy to try to squeeze in people that don't fit the mold of what they would. Oh, you're going to ruin the whole average if you admit that person. Right. Crap on that.
Rich, what do you have?
My pick is a fairly local. I will elaborate. children's museum called the Science Mill. So this is a local pick, and I picked its website, and I also added to that a short video, thanks to Kathy and Nels, that gives a sort of highlights what this is. So this has Nels picked this on.
No,
I think so.
You think he has?
That's why I've heard
that's
about.
right. The Science Mill. He did. Let me take a look while you talk. Yeah.
So this is a small, now currently, science museum oriented towards children that is in Johnson City, Texas. That's where Lyndon Baines Johnson grew up. It takes me about almost an hour to get there, and I live in South Austin, so it's probably about a similar amount of time because it's westish from downtown Austin. So it's in the middle of kind of nowhere, but it's beautiful country. I mean, the drive down there is awesome. And this was set up as kind of a retirement project by Nels's father and Nels's stepmother. Okay. And
cool.
At least according to Nels's father, and from what I can read, it's his stepmother, whose full name I don't know. Her first name is Bonnie. And I'm sorry, but I forget her last name. But we'll come back with that sometime.
Bonnie Baskin.
Bonnie Baskin. Thank you. I think she's really the prime mover on this thing, and she's absolutely passionate about bringing science education, STEM education, in particular to the underserved children. So this not only is a small museum facility, but they also do a lot of outreach. They have field trips that come to them, and they also send people out to schools to do stuff, and they're really doing an amazing job. The reason this is really great, they went to Johnson City, I think, as a retirement place for other reasons, and this is an old defunct grist mill. That they have converted into this science museum. So it's entertaining looking, and they've made use of the whole facility and have a number of little exhibits. They just opened an aquaponics exhibit, and Nels came out from Utah for that. They had an opening and a fundraiser at the same time, and Nels invited me and my wife, and it just so happened that Harper, who of course is a TWiV celebrity, my granddaughter, was with Ibby and I at the time, all of the other Maines family doing other things. So me and Ib and Harper went to this opening, and it was a blast. And this aquaponics exhibit is really terrific. This is great, and I would really like to see it grow. And as a matter of fact, on Wednesday after Thanksgiving, I'm going to go down again and meet with Bob, and we're going to discuss some ideas, and maybe this will be a little pastime for me.
Well, cool. Yeah, Nels picked it on TWiVo number 6.
There you go.
A long time ago.
Wow. Makes perfect sense. Yep.
The video is really cool. It has testimonies from the children who've been there. One of them from early on, and then several years later, there's a camp as well, a summer camp that some of them go to.
Right.
Cool.
Yeah.
Dickson, what do you have?
Strangely, in order for me to be reeled in, so to speak, by reality of the truth, I'm more than willing to mea culpa with the naming of the 12 cranial nerves and a teaching anatomy video to show you how to remember all of that stuff and to actually teach you what each one does. And it's quite important to know all of this. So sorry that I mistook.
Okay. No.
Misspoke.
We all make mistakes.
But of course,
and we learn from our mistakes.
No, tell me about it.
With this.
No, that's right. That's great. Right. So this is just a gentle reminder of here they are, and deal with it.
It says you have a mnemonic, right?
Yeah, but
and then it's not all politically correct.
By the way, there were a couple of them that I thought putting in there, and I didn't do it.
Okay,
right. I posted the mnemonic and then this link to some other things that it's standing for, or other mnemonics.
I should have known it's in purple.
Mm-hmm.
Some of the some of the medical school mnemonics
are
quite.
Kathy, what do you have?
I picked the National Academy of Sciences report on sexual harassment of women, which is available as a free PDF. And a couple things about it. I also picked the infographic, which is an iceberg. And the original iceberg graphic was designed by someone at the University of Michigan. So this National Academy's report came out, exactly, I'm not sure how long ago it came out, but pretty recently. And it was based on a 2-year project. And they rolled it out here in 3 different seminars. I went to the first or second one. 3 people from the University of Michigan were on the 21-person panel, and 3 people from Berkeley, and then a number of other people. And so some of those outside people came to the rollout that I went to. So I'm trying to find where I linked to it. There it is. A couple things about it. It's quite a long document, 313 pages, but there's a preface which has a couple of points. So the Committee on Women in Science, Engineering, and Medicine developed the idea for this more than 2 years ago. And through the work, it became clear that sexual harassment is a serious issue for women at all levels in academic science, engineering, and medicine. However, we are encouraged by the research that suggests that the most potent predictor of sexual harassment is organizational climate. This means that institutions can take concrete steps to reduce sexual harassment by making system-wide changes. Based on the strength of the research, we're optimistic that academic institutions, campus-wide as well as within schools, programs, and departments, can meet the challenge of reducing and preventing sexual harassment. Okay, so that's from the preface. And then I'm scrolling down now to the summary. So there's sort of an executive summary. And I highlighted a couple of things that the academic workplace has the second highest rate of sexual harassment at 58%. The military has the highest at 69%. And too often, judicial interpretations of Title IX and Title VII have incentivized institutions to create policies and training that focus on symbolic compliance with current law and avoiding liability, and not on preventing sexual harassment. So they talk about 3 categories of sexually harassing behavior, and 2 of these are captured in the iceberg picture. So gender harassment, unwanted sexual attention, and sexual coercion. And so the iceberg, if you look at it, the top picture, or the top part above the water, is sort of the most overt. And the way that Lilia Cortina, who designed the original figure, talks about it, she says this is the come-ons, the actual coercion and so forth. And then everything under the water is gender harassment, verbal and non-verbal behaviors that convey hostility. So even just clicking on that, I think, is really informative. So those are the 3 categories. Then the next thing they talk about is 4 aspects of the scientific, engineering, and medicine workplace that tend to silence targets of harassment and limit career opportunities for targets and bystanders. And so there's those 4 points, and then there's a number of recommendations of how to overcome this. And so I think if you just were to download this PDF, as I said, it's free. The summary is not that long. And what I'm trying to convey is that while it's a serious and potentially very worrying situation, the report points out a number of positive things. And what came out across to me from the rollout is that individual universities and units are going to need to work on something, that's sort of customized for their own situation. They did not want to prescribe things for everyone. So it's, I think, a really important report that everyone should take a look at.
Yeah. And just if you go to the National Academy's page and you look at this, you'll see the buy a paperback or buy the ebook staring you in the face. But the slightly more subtle, smaller type download free PDF in the upper right or read online. So you do not have to pay for this.
Yeah.
And the only other thing I wanted to point out is the figure, this excellent graphic that Kathy linked to, it actually covers all three categories. It's just divided up into the unwanted sexual attention is this triangular that's partly above and partly below the water.
Right. And I forgot to say, so the top one is the come-ons, and
coercion,
the stuff under the water is the come-ons and then the put-downs. So that's an even faster way of remembering that. And actually, this is not Lilia's original diagram. She's not quite as fond of how they modified her diagram, but she hasn't published hers yet. So, yeah.
Did anyone see the evening news last night? Just last night. Dartmouth University.
Oh, yeah.
Science, not the general faculty, the science faculty.
Mm-hmm.
So those, you know what that does to a university, of course, right? I mean, the reason why they try to squash all this stuff is because people don't want to go there afterwards, right?
That does much more than the U.S. News.
Exactly.
Business.
Yes. Mm-hmm.
My pick is David Quammen's latest book, The Tangled Tree. Terrific history of trees of life and tracing from before through Darwin and to the modern day with Carl Woese and the discovery of horizontal gene transfer and how that influences the structure. I'm not going to say much more because Nels and I interviewed David yesterday on TWiVo. It'll be up Saturday, number 37. You can hear all about that from him.
Wonderful.
Cool.
Great. Very good.
And that will do it for TWiV 520. Please subscribe on your favorite podcast player. Send us your questions and comments, twiv@microbe.tv, and consider supporting us financially, microbe.tv/contribute. Dickson Despommier is at thelivingriver.org and parasiteswithoutborders.com. Thank you, Dickson.
You're welcome, Vincent.
Brianne Barker is over at Drew University. On Twitter, she is BioProf Barker. Thanks, Brianne.
Thanks. It was great to be here.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thank you. This is a lot of fun.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove is at turbidplaque.com. He's on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
Now, Vincent. Yes, sir.
We have to wish everybody a happy Thanksgiving.
Mm-hmm.
Okay.
Sure.
When do you want to do
an
that?
entire list. I do it right now. I just extend my happy Thanksgiving to everybody listening and to everybody on the show.
Happy Thanksgiving.
We're a little late for the Canadians, but belated happy Thanksgiving.
Right, exactly.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASM for their support and Ronald Jenkins for his music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Hello, Alan.
Hello.
How are you?
Alan.
Doing okay. How about you?
All right.
How am I doing?
Yeah, right. Well,
Hey, Ed.
we're going to work on the pronunciation of that.
Well, what is it? I thought it was Coke. Kathy, is it really cock?
Koch.
Oh, the cock.
Yeah, that's what I said. Koch.
If you put it into Google Translate, it's the word for cook.
Koch.
Right.
And you can push the little translation thing.
Koch.
Koch.
Not a hard. Well, it depends what part of Germany and High German, Low German, but Koch.
So throughout Germany, they will go Koch of some kind.
Kind of like caw, like a crow. Koch.
There you go.
It's too subtle for Americans, right?
Much.
Well,
It depends on the American.
Yeah, I've heard it as Coke and said it as Coke, but I guess it should be Coke.
That would be way wrong. That would be like saying
Coke is wrong. It's totally wrong.
It's totally wrong.
Like saying Van Gogh.
Right.
You know,
Well,
where the Dutch say it differently.
Yes.
Well,
the British.
the Coke Industries, they're probably mispronouncing it, right?
Of course they are. And Ed Koch was mispronouncing it.
Well, they can pronounce their names however they want. But
However they want, right.
And the Koch brothers. What about the Koch brothers?
Yeah, that's what we did. So there are plenty of Italian names where they're Americans. They don't know anything about Italy, but they have an Italian name and they butcher it. But you talk to them and they say, that's the way I want to do it. There's a well-known HIV virologist who calls himself Bob Silicano. No, there's an I in his name. It's Siliciano, but he doesn't pronounce it that way.
Right.
And probably his parents didn't pronounce it that way, and so forth. It's lost, which is sad, but it's up to them.
Yeah.
Look who's talking.
There's a podcaster I listen to. Her name is, she calls herself Megan Maroney. It's Maron. Maron.
And you don't say the final E? Marone?
You don't have, it depends what part of Italy, right?
Oh, because in sung Italian, you would say all the vowels. So
Marone.
I think.
You wouldn't say Maroni. No,
you would say Marone.
Yeah. Yeah. So you have to respect what people want to call themselves, right?
Yep.
Right.
But having been German, I think we can presume that the Koch we're talking about would have been Koch, right?
Right.
Koch.
Yeah.
Yeah.
My father used to
like
call
Spindler.
My father like Spindler.
Fräulein Spindler.
Fräulein Spindler.
Michael Spindler used to be CEO of Apple.
Yeah.
You related?
None.
Nicht. My father used to pronounce our last name Despommier.
Aw.
And, until I got out of the house, that's the way I pronounce it.
Now you say Despommier, right?
Despommier. It's Despommier, yeah. It is.
That's good. All right. There's our
here we go.
Our little after Easter egg.
All right.
Koch.
Koch.
Oh, it's
Koch. That's what I said. It's like kind of like a crow cawing.
Koch. There's an awful lot of saliva here. Rich, are you there?
Microphone covers are.
I'm here.
You like to just
I'm just listening, enthralled to this, trying to avoid driving into an abutment.
Right. Right.